AP Biology Notes on Photosynthesis. Continue with the discussion of C4 and CAM plants. So in hot arid climates, there is a problem with dehydration for plants. And sometimes you have to trade things off, okay? And photosynthesis falls prey to this in a lot of places. When it's hot and dry, the plants are going to close their stomata, which is going to help conserve water. But by doing that, it's also going to limit photosynthesis because you need to have access to carbon dioxide for photosynthesis to continue, and you're going to have a buildup of oxygen. And so this is going to create or it's going to favor a wasteful process, or apparently wasteful process, called photorespiration. In photorespiration, okay, you're going to form 3-phosphoglycerate using rubisco. But in photorespiration, rubisco is going to add oxygen rather than carbon dioxide in that process, and it's going to produce a 2-carbon compound. It's going to use the oxygen, the excess oxygen that's building up, and release carbon dioxide, but it's not going to produce any ATP or sugar. So it seems to be a wasteful process. It could be because rubisco first evolved when there was no oxygen, or very little oxygen in the atmosphere, and a whole lot more CO2. And so what happens in photorespiration, it seems wasteful, but it also limits the damaging products of the light reactions that can build up when the Calvin cycle is not present. In plants that live in hot, dry climates, it can be a problem. Photorespiration can present a problem because it's going to use a lot of the carbon that is fixed by the Calvin cycle in this more wasteful process that does not produce sugar. So there are some plants that have developed alternative methods of dealing with this. Some of them are called C4 plants, and C4 plants are going to reduce the cost of that wasteful process by incorporating the carbon dioxide into a 4-carbon compound in some of the mesophyll cells, and it's going to be catalyzed by this PEP carboxylase, which has a stronger or higher affinity for carbon dioxide, and it can fix carbon dioxide even when there's not very much carbon dioxide present. And then these 4-carbon compounds are exported to cells called bundle sheath cells, where they're going to release the carbon dioxide that is used in the Calvin cycle. So rather than using up the oxygen and releasing more carbon dioxide, it's going to save the carbon dioxide to be used in a slightly different way. So here we have a different anatomy in the C4 leaf. Okay, we have our mesophyll cells where photosynthesis is going to occur, or where the light reactions are going to occur, and then we have our bundle sheath cells that are inside that. Okay, and so here's what happens in that pathway. The carbon dioxide comes in and is catalyzed by a PEP carboxylase, undergoing a couple of intermediate steps into oxaloacetate malate, both of which are 4 compounds, should be familiar to you from respiration. Okay, and then those can be either changed into pyruvate and go back up here through this cycle, or they can release carbon dioxide in a gradual way to provide carbon dioxide for the Calvin cycle to produce the sugars that are then passed into the vascular tissue. So we have a little bit different arrangement of cells, a slightly different pathway, but it saves the plant from having to keep its stomata open during the day, and when it's hot and dry, and prevents the photorespiration process from continuing. Now, in the last 150 years or so, the carbon dioxide levels in the atmosphere have risen greatly, and so the C3 normal plants and C4 plants are going to be affected differently as a result of that. And so we may see some changes in populations of plants related to that because of the change in atmospheric conditions, which can change the weather and produce in some areas more hot, dry days, and in other areas more cooler, wetter days. And that could definitely change the percentage of the C3 and C4 plants in different locations because of this, and it could be a concern, one of the many concerns involved with global climate change. There are other plants called cam plants. Okay, this includes things like succulents, or succulents include things like cactus, and they use a different kind of metabolism called crassulacean acid metabolism, which is more easily called CAM, to fix the carbon. In CAM plants, the stomata are open at night, and at night the carbon dioxide is incorporated into various organic acids, and then during the day the stomata are closed and the carbon dioxide is released and used in the Calvin cycle. So here we have two different kinds of carbon fixation that can occur. Plants like sugarcane, which are C4 plants, okay, the mesophyll cells are going to change the carbon dioxide into an organic acid, and then the carbon dioxide is going to be released in the bundle sheath cells where the Calvin cycle will occur, separating the steps in the process by space. And in CAM plants, we have a temporal or a time-related separation of the steps in which at night the carbon dioxide is incorporated into organic acids or fixed into organic acids, and in the daytime the organic acids can then release the carbon dioxide to complete the Calvin cycle and produce the sugars. In both cases, we still have the production of sugars by this photosynthetic process, but we have, in the types of climates that these different plants live in, we have a different way of doing it to help prevent photorespiration, which can be harmful. Now, a review of why photosynthesis is important, of course. The energy that enters the sunlight gets stored as chemical energy. The sugar that's made in the chloroplast provides chemical energy to synthesize other organic molecules in cells. The plants store excess sugar as starch, and then can be used later by the plant or also by animals that eat the plant. And then it also, in addition to making our food, it produces the oxygen in our atmosphere. So photosynthesis is an essential process. It's necessary for life on Earth. We could not survive without it. And just as a quick review, here we have a summary diagram showing the entire process, the light-dependent reactions in photosystem 2 and photosystem 1, and their associated electron transport chains split water, release oxygen, produce ATP and NADPH that are then used in the Calvin cycle along with RuBP to produce our G3P, which produces sucrose. It can also be used to make starch for storage. We're using the carbon dioxide here, producing NADP plus and ADP that are then reduced in the light-dependent reactions back to their active form.